Hello, folks. In this lecture, we're going to talk about kind of a fun article for me. This was my first publication that I ever got. This was my senior honors thesis. So I was an honors student when I was completing my bachelor's degree, and this was my honors project. And what we were looking at was episodic or episodic-like memory in pigeons. And I wanted to talk about this one for this chapter because obviously we're focused on learning and memory, but I really wanted to talk about this one because a lot of the animal research that we've talked about in class, a lot of the non-human animal research, has been really focused on looking at brain structures and things like that. And this research that we're going to talk about in this lecture is more behavioral. But again, it is focused on episodic or what looks to be episodic-like memory in pigeons. And it's a fun experiment, and I wanted to share it with you all. So I hope you like it. Okay, so let's take a look at the types of memory. We can see a similar diagram in our book chapter for this material, but if we're looking at types of memory, memory is typically broken down into short-term and long-term memory. Short-term is also kind of discussed, I think, more frequently now as being working memory. The type of memory that we're going to focus on here is a form of long-term memory. Specifically, it is a branch off of explicit memory. So if we're looking at long-term memory, you have both explicit and implicit memory. Implicit is unconscious memory, so it's very much procedural, how to do things like riding a bike, I think is a really good example. You don't really think through the breakdown of how you ride a bike, you just do it. So these are the type of things that would fall into unconscious memory. Also, classical conditioning would fall under this as well. But explicit memory is memory that we say that you are conscious of. So if we look at a branch off of explicit memory, we have declarative memory. So that is memory for both facts and events. So memory for facts is semantic memory. This is just basic concepts and factual pieces of information. So for example, who's on the $1 bill? So that would be a fact. Now, when you learned who's on the $1 bill, that is an event, and that's different. That would be an episodic memory, and that is going to be the focus of our lecture today, or the focus of this article. So episodic memory is a branch off of declarative memory, and it is really focused on your recall of events from your past. So it's got to be an event that happened for you in your past, and it is something to do with your experience. Now again, just to reiterate, episodic memory is different than semantic memory. Remember, semantic memory is just factual stuff. That's just your ability to retain factual information. It's not when you learned it or how you learned it, it's just the fact that you know this piece of factual information. Another example that I give often is who was the first president of the United States. Whereas episodic memory is your ability to recall aspects from some past personal event within your lifetime. So for example, what did you do on your 16th birthday? You could tell me, did you have a party or not? Did you get your driver's license? Did you get a new car? Like, what happened to you on that day? Your ability to kind of go back in time and relive that event of your 16th birthday, that's an episodic memory. Quickly, let's take a look at some of the brain areas that we think are involved with episodic memory. So one thing that seems to be an agreed upon point is that structures within the medial temporal lobe are needed to be able to form new episodic memories, specifically the hippocampus. So if we're taking a zoom and look here, these areas here would be what we think to be most important, but also these related areas as well. So also other areas of the medial temporal lobe. So that would be these cortexes here and here. And some people also believe that the hippocampus actually stores memories. There's kind of debate back and forth about that. Some newer research has suggested that the hippocampus stores memories, but only for a brief period of time. Nonetheless, though, the hippocampus seems to play a very large role in episodic memory. And if there is damage to this structure, then episodic memory is severely affected. A lot of times what will happen is that people may be able to perform a behavior for example, that they would have learned previously, but they have no recollection of how they learned how to do that behavior. So they can do it, they just don't remember actually acquiring that behavior before. Another area that we think is heavily involved is the prefrontal cortex. So that, of course, is this area right here. We think that this particular area is involved in more, if you want to think about it as executive functioning, that's an acceptable way to think about it. Really, we think it may be just involved in the monitoring of episodic memories and the management of them. And last is, the last one that I wanted to mention here is the inferior parietal lobe. So that would be this area right here, this yellow area here. More recent research has suggested that this particular area may be involved in that feeling that you get when you're reliving an event, and it's kind of the sense that you get that memory is old, so that it's in the past. It seems like it happened a long time ago, that it's not now, it's in the past. It feels like an old event or an old memory. We think that this particular area may be involved in that feeling. Okay, so let's look at some ideas of how to study episodic memory. In 72, Tulving proposed that episodic memory is just the basic process of encoding and retrieval from memory of unique past personal experiences. So this was the basic working definition of episodic memory. Now, please note that the definition is very specific about being past and personal experiences for the individual. More specifically, within Tulving's definition, he proposed that a person who has episodic memory should be able to recall three things. They should be able to recall what happened during the event, when the event happened, and where it happened. So what, when, and where. Now we're going to look at a more recent study that was done with scrub jays. Now, these little jays are what we call food caching, and what that basically means is that they can store food for long periods of time and then go back and retrieve it later. Now, as you can imagine, if they're going to hide food, what needs to happen is that they have to be able to have some memory of where they hid the food so they can then go back and get it later. If they don't remember where they hid it, then there was no point. So Clayton and Dickinson wanted to do an experiment with these food caching scrub jays to test whether or not they exhibited episodic or episodic-like memory according to the Tulving definition of the what, when, and where. So these jays were trained to cache either peanuts or waxworms, and this would have been the what part of the episodic memory. So peanuts or waxworms. So first, we need to recall what was cached, and then also where. So they were allowed to cache these peanuts or waxworms in ice cube trays. They had various ice cube trays set out, and then the jays could choose where in each ice cube tray that they wanted to cache these items. And then this one was quite important, the when. So the rule here would be, if these scrub jays were allowed to go back and retrieve their food item that they cached within a day's time, they should go get the worms because they would still be good. But if they were allowed to go back 4 days later, they should go get the peanuts because the worms won't be good anymore. They'll be bad at that point. They should retrieve the peanuts. And the test is, do the jays remember where they cached the item? And given the time frame that they're allowed to go back and retrieve the item they cached, do they go get the appropriate item? And the answer is that they did. They did. So in that experiment, the jays were able to retrieve the appropriate item given how long the time frame had been from when they had cached. So if it had been a day, they retrieved the worms. If it had been 4 days, they retrieved the peanuts. And so the conclusion from the Clayton and Dickinson study was that scrub jays seem to have what looks to be episodic memory. Now, that brings us to this question here that we're taking a look at. Does this demonstrate episodic-like memory? That is being able to recall what, when, and where about some event, enough to say that you actually have an episodic memory of that event. And the laboratory that I was in grad school was really arguing at the time that this really wasn't sufficient. So being able to recall what, when, and where about an event wasn't enough to demonstrate that the memory for you was an episodic memory. And then also, there could be certain instances where you may not remember all three components, what, when, and where, but it's definitely an episodic memory for you. So I'll give you a few examples. Let's think about it not being sufficient. So I could tell you what, when, and where regarding the Declaration of Independence. I could tell you what it is, where it was signed, and when it was signed. But that's not enough. Obviously, that's not a past personal event for me. Guys, I might be a little bit old, but I am not that old. I was not born in 1776. I was not around then, okay? So that's not sufficient, obviously. Now, the other component, maybe it's not necessary. Have you ever been thinking back to a time, a specific event in your life, and you knew what was going on, you know who you were talking to, you know where you were, but you couldn't remember when exactly it was? That happens all the time to us. But we would argue that that's still definitely an episodic memory for you because it is past, it is personal, and you can actually kind of travel back in time and relive the event. The key here is that we want to be able to test subjects to see whether or not they exhibit episodic like memory in such a way that they actually have to go back in time and relive the event and experience an episodic like memory versus just basically learning rules of what, when, and where. So again, with the Declaration of Independence example, I can learn the what, when, and where there. And if you were to test me on it, I can tell you what, when, and where about that event. But it obviously isn't a past or personal event for me. So it's not an episodic memory for me. So how do we test these subjects in a way that we can ensure that they aren't answering the questions that we're asking based upon rule-based learning? They're actually having to kind of go back and relive the event to answer the question appropriately. So within my lab, in order to be able to kind of try to get at whether or not, specifically for us, pigeons could experience episodic memory or use something that looked like episodic like memory, we proposed that at the time the question being asked, whatever the question was, needs to be unexpected, because if you're expecting to be asked something, you can prepare an answer. And that answer then becomes semantic or just a factual piece of information for you. But if you don't have time to prepare an answer and the question is unexpected, then that will kind of make you go back in time and relive the event to be able to answer. And that was our goal with our pigeons. And I'll give you an example here. Let me ask you this question. What did you have for breakfast this morning? You probably weren't expecting that I was going to ask you that. So you probably sat there for a second, thought about your morning, had to kind of go back and relive your morning and think about, did you wake up on time? Did you wake up late? Do you always have the same breakfast every day, or do you have something different every day? You know, what did you do this morning and what did you actually have for breakfast? Now, you probably had to go back and relive your morning to answer this question because you weren't expecting that I would ask you that. Now, if I ask you this question every single day for the next few days, by the third, fourth, or fifth day, you're going to automatically think as you're eating breakfast, Jess is going to ask me what I had for breakfast today. So I should look down at what I'm eating and remember, okay, I'm eating a Pop-Tart. That's what I'm having. That way, later that day, sure enough, I ask you, what do you have for breakfast? And you just tell me, I had a Pop-Tart. Something changes on that particular day because now you're not having to go back and relive your morning. You actually had a prepared answer for me. You expected me to ask you that question. So your breakfast is now kind of a semantic memory for you. This is what we wanted to avoid when we were testing our pigeons. We wanted to make the question that we asked them unexpected so that they would have to kind of go back and relive the event in order to answer the question that we were asking correctly. Now the question we wanted to ask our pigeons was, where did you last peck? So we wanted to ask them, where was their last pecking event? Where did it last occur? Now this seems like a pretty lofty goal. Like, how do you get to this point with pigeons? It takes a little bit of time. We worked in 3 phases. The first phase was what we call language acquisition. So that's really just training the birds how to report back to us and how to peck the keys and what each of the keys represents or what each key does. And in phase 2, we just give them more experience pecking and reporting back to us. And then in that final phase, that third phase, that's when we actually tested them with the unexpected question. So most of you all have seen this before. I'm sure I showed you guys this at the beginning of the term, but this is a pigeon in an operant chamber, and this is the exact type of operant chamber that we used for this task. And these were the type of pigeons that we used. This is a white Carno pigeon. These guys are much larger than the ones you see outside. These are taller and bigger, just in general. So they're actually quite tall. This is actually a taller operant chamber than what you might think. But within the operant chamber, you can see there are 3 response keys here. And this is where we present to them either colors or shapes depending upon what it is that we're trying to achieve. And the pigeon can peck those, and if we want to reinforce a certain response, then we reinforce or give food here through a hopper. So there's actually a hopper back here and a food magazine, what's called a food magazine, that holds the seed here. So the hopper will swing up and a light will turn on and the bird can come down here and get food. Okay, so let's take a look at what the language acquisition phase looked like. So this is phase 1. So here we're looking at our 3 response keys. So what we would do is present a white light either on the left-hand side or the right-hand side. And here we have, for example, the white light is being presented on the left key. The bird would have to peck this once, and then this would shut off. So 1 peck, or an FR1, if you've had a learning class, a fixed ratio of 1, so fixed number 1 peck. And then that would shut off. And then we would have a shape stimulus triangle that would appear in the center, so the center key. And this really is to move the bird away from the side key. So if the bird was pecking here and this light extinguishes, then they have to shift over to the center and peck this. So then after they peck this center triangle stimulus, and again this is also an FR1, so 1 peck, it also shuts off. And then we present the birds with a choice, red and green. And this is the equivalent of where did you last peck? And the rule here is if you pecked on the left, so the white light in the first part of this trial appeared over here on the left. Here, when they're getting the question of where did you last peck, they would need to respond to the red key. And if they did that, they would receive food. And if they responded to green, they did not get food for this. And again, red and green in this case can be viewed as the question of where did you last peck. So as you can imagine, for a bird in this condition, left would mean they would need to peck red. If they received the white light on the right, then they would need to peck green. And I will go ahead and make sure that you know that we did what is called counterbalancing. So what that means is that we presented red and green on either key. So this question could be asked in the way of green being here and red being here. And that would happen on half of the trials each way. So we would alternate where we presented the red and the green. And also we did have another group of birds that had to respond to green for left and red for right. So we balanced all of the presentation and also what color the bird had to respond to report to us what location they pecked at. So the basic rule here is if I pecked left, then I need to choose red. If I pecked right, then I need to choose green. And again, this is the question of where did you last peck? The pigeons learned this very quickly. But as you can imagine, they're getting trial after trial after trial of this. So this is not unexpected because every trial looks like this. But keep in mind, they have to learn there has to be learning of this rule and how to report in this way before we can ask it in an unexpected way. So in phase one, we're training them as to what these stimuli represent so that when we give them these stimuli later in an unexpected way, they'll at least know how to respond. So we give them several trials of this. They acquire this pretty quickly, and we're performing well above 90% here. So well above 90% correct on these trials. And so once they got that experience with phase one, we then moved them with phase two. Okay, now we're on to phase two. Now keep in mind, we don't want that "where did you last pet" question to be expected when we're going to test them. So we have to build our probe trials, which are coming in phase three, in such a way that we can ask that question in an unexpected way. And that's really what phase two is going to help us do. So phase two is very different than phase one, and that we're not asking our pigeons here to tell us where they last pecked at all. We're having them do basically a symbolic matching task, and I'll show you what that looks like. So on these trials, the pigeons would get one of two sample stimuli. They would get either a blue light or a yellow light. And here in this trial, this is obviously a blue light trial. So they would get a blue light on the center key, and they would have to peck that. And then after that, we would give them either a choice between vertical or horizontal lines. And for a pigeon in this particular condition, vertical would be the correct choice if the sample stimulus was blue. So this is just a rule learning task basically here. So if I see blue, I choose vertical. That's it. That's all they have to do here. And then after they peck the line stimulus, so let's say this bird was correct, it chose vertical. After that, an orange light would be presented on the center key. So here's kind of the stages of the trial. So let's start from the beginning again. The bird would get a blue light, they would peck that one time, and it would shut off. All the keys would go dark. Then the bird would be given this choice here between vertical and horizontal, and let's say that bird chose vertical. These lights would then shut off. And then the center key would be lit up orange. One peck to this would either result in food or no food, depending upon if the bird chose the line comparison correctly. So our bird in this example, he chose correctly, he chose vertical, so he is going to get food after he pecks orange. So on the other half of trials, the birds would get a yellow light. So here we have the yellow light on the center key. Once they peck that, it would shut off, so all the keys would be dark, and then we would present the line stimuli, vertical and horizontal. And you probably already guessed it, for this bird in this condition, horizontal would be the correct response following yellow. So let's say our bird does a good job, he chooses horizontal, then all those keys shut off, and then again the center key is lit up orange. He just needs to peck it one time, and then he will get food if he was correct, and our little guy was correct, so he's going to get food here. So again, this is a symbolic matching task. So there's a symbol that is the correct choice following a color, right? So here the basic rule in this task that we just saw is, for blue, choose vertical, for yellow, choose horizontal. And I'm sure you're wondering, what about this orange thing? Like, why are they having to peck the orange key? You'll see here in just a second. All right, that brings us to phase three. Now remember, we want that question of "where did you last peck" to be unexpected. So we're going to try to present that question in a way in phase three that is novel, that they've never seen that before presented to them in that way. So basically they have experience with the question itself, but it's never been presented to them following this certain task. So let's take a look at what that looks like. In phase three, these are what the probe trials would look like. So when we would ask them that unexpected question. So if you notice, right off the bat, it looks very much like phase two. In fact, it looks identical to phase two. So we have a blue light, And just like in phase 2, they would peck the blue light, all the keys would go dark, then they would get their line comparisons, and again, for our little guy, vertical would be correct following blue. So let's say he's doing really well, as most of the birds were at this stage, choosing correctly. So our little guy chooses vertical, everything shuts off again, and then that orange light comes on in the center, and our pigeon pecks that key, that orange key. Now at this point, if it were to be like phase 2, our pigeon would be getting reinforced right after packing orange, because he chose his line comparison correctly. But instead of that, we present the question of where did you last peck? And at this stage, this question has never been posed after the orange key. So this should be unexpected. Now, if we track back to phase 1, in order to report that the bird had pecked left, they would need to choose red. So red would technically be marked as correct here, because our little guy, remember, chose vertical correctly. So here, when given the question of where did you last peck, left or right, red should be the one that is selected. We did this for yellow sample trials as well. So let's say we have a yellow trial. The yellow light comes on, they select yellow, it shuts off, line comparisons come on. In this case, horizontal is correct. And then the orange light comes on, the pigeon pecks that, and then we pose again our unexpected question. But in this case, if they chose right, which again most of the birds would have because they were doing so well with this task, green would be the correct choice, because green would be reporting that they had last selected right, the right key. Now think for a second here. Why wouldn't we just give them red and green right after the line comparisons? Why have this orange key here? I'll give you guys just a second. Okay, let's think about it. Let's take a look over here. Let's say that our pigeon is standing in front of this key, and their beak is obviously on this key because they pecked it. If we were to present this question, this red and green, symbolizing where did you last peck, directly after they just pecked this, then when these stimuli come up, their beak is still on this left key. So they could use their body position or their beak position as a cue for where they last pecked. And in order to try to prevent that, we had them move to the center key and peck this orange key. This again is to control for any body position or beak position cues that they could use to inform them about which response would be correct here. Now, this was our best attempt at trying to present this question in a novel way. During phase 3, we had a mixture of phase 1 and phase 2 trials, because we don't want our birds to forget what this means. And we know if we go for a very long time without presenting them with phase 1 trials, so the white key lights, and then they just have to tell you where the white key light appeared. If we don't give them continued experience with that, then we could just have an issue here where when we present them with this unexpected question, that they don't do so well, but it's not because they can't kind of relive their last pecking experience, if you want to view it that way. It could just be that they don't remember what to do when they get this question. So we have to give them some phase 1 trials throughout phase 3. We also give them some phase 2 trials that just terminate right here after this orange key. So out of 104 trials in a phase 3 session, only 8 trials looked like what we're seeing here on the screen. So we only had 8 probe trials. Now I will go ahead and mention too, we used on these probe trials here something we call non-differential reinforcement. Basically what that means is that we reinforce them half of the time regardless of what they did. So let's say our little guy was correct all the way through. So here he chose vertical, and then down here he reported that he pecked left by choosing red, which we would mark as being correct. but half of the time he wouldn't have gotten food for that. Half of the time he would have gotten food for that. And basically, we didn't want to train them how to respond on these probe trials. So we really just wanted to see, could they do this better than chance or not? And here we're looking at performance on probe trials. We're looking at subject numbers, so individual subjects here 1 through 7, and we're looking at percent correct above chance. And overall, as a group, if we average performance out, these birds were better than chance on probe trials. So what we can conclude from this study is that just overall, this brings us closer to being able to observe what, by a fair number of people has been thought to be a purely human ability in a non-human species, even better, a non-primate species. For funsies, I wanted to show you one more article. This one was done with rats. This is a picture of Jonathan Crystal. This is a researcher that I've known for longer than I'd like to admit at this point. This is a researcher from IU. He attends a conference that I go to annually, and this was done by his graduate student. And it's fun, we were actually graduate students at the same time, Wendy Zhao. So in this article, what they did was they put rats in a radial arm maze. So in this case, it's an 8-arm maze. And they did something called first helpings and second helpings. So in first helpings, rats were allowed to go down 4 arms of the maze, 3 of which had regular rat chow, and then 1 had chocolate, which if you don't know, you can already guess, rats really like the chocolate arm the best. They like the chow just fine, but they're all about that chocolate arm. So first helpings, this is what it looked like. 4 of the 8 arms were accessible. Of those 4 arms, 3 had chow and 1 had chocolate. The rat would then be taken out of the radial arm maze for a bit, in this case a 2-minute delay, and then they would be placed back in. And at this point, they would be given a retrieval cue. So for example, if the central hub here, if it had chocolate pellets in it, that was a signal to the rat that the chocolate arm was going to replenish. And so what the rat should do is revisit the chocolate arm if they got this signal here, but not if they didn't get that same cue. So if they didn't get the chocolate in the central hub, then they really shouldn't bother visiting the chocolate arm again because it didn't replenish. So during second helpings, following the retrieval cue, this is what it would look like. So for the rat that got the signal that, hey, there's chocolate in the hub, that means that the chocolate arm is going to replenish. They could get chocolate down the chocolate arm. But for the rats that did not get that chocolate in the central hub, the chocolate arm did not replenish. So here what they were looking at was basically, did rats revisit the chocolate arm more often if they got this cue? And the idea is that they would have to kind of recall where the chocolate arm was before and whether or not they got chocolate there previously to then be able to figure out how to use this cue. And what they found in this particular condition was that rats did revisit more often when they got that replenishment cue versus when they did not get the replenishment cue. In a follow-up, they did an experiment where they basically did the exact same thing, but instead of a 2-minute delay, they did a 1-hour delay. So this was more trying to test long-term memory. And again, the same exact design, except a 1-hour delay between actual experience in the maze and then presentation of the retrieval cue. So other than that, again, everything looked the same. And again, they had a very similar finding here. Rats were much more likely to revisit the arm if they got the replenishment cue of chocolate in the central hub. So it seemed as though they were able to at least recall where the chocolate arm was and revisit that arm more likely if they got that replenishment cue than if they did not get that replenishment cue. So continuing on, they continue to use a radial arm maze, but they tested their rats in 2 separate rooms. So they put their rats in a radial arm maze in test room 1, and they blocked off 4 of the arms. and they baited the other 4 with chow. So they allowed the rat to go down these 4 arms and retrieve the chow. And then they would remove the rat from the maze, take them back to the colony room, and then take them into a second test room with a second identical radial arm maze. Now they would bait the other 4 arms that had not previously been baited with chow in room 1. They would bait those with chow, and they wanted to see if rats would travel down the previously blocked off arms to retrieve chow. That is, could they recall which arms they didn't go down before to retrieve the chow? And they found that the rats did differentiate between the 2 rooms, and they were able to travel down the previously blocked off arms much quicker than they did the arms that they had already depleted in room 1. Right after this, they followed up with using the same test room 1, test room 2 idea, but in this case, they baited one of the arms in test room 1 as a chocolate arm. And then they would remove the rat from this apparatus, again, take them back to the colony room, and then take them into the test room 2. They would give them a retrieval cue. So again, this is revisiting back to their first experiment where they would give a retrieval cue of chocolate in the central hub or nothing in the central hub. And remember, this cue, while the rats have seen this cue before, it's never been used in this context before. So what this is trying to tell the rat is this chocolate arm that you saw in test room 1 is going to replenish if they're in this condition here, and that's the cue for that. So that is the cue for the rat to recall which arm had the chocolate in it. And if they're able to do that, then we should see rats revisiting the chocolate arm more readily in this condition here. And in second helpings, that arm does replenish if they got that retrieval cue. And again, rats, if they can recall what arm was the chocolate arm, they should travel down this arm quicker, or they should be more likely to go down this arm than rats that don't get that same cue. And that's exactly what they found in that particular condition when they did that chocolate probe. The revisit to that particular arm was much higher than what it would be by chance. So that's all I have for you guys in this PowerPoint. I just really wanted you guys to see something that, see multiple things actually that have been done with non-human subjects and even further non-primate subjects, looking at episodic like memory. This is just some of the work that's been done. So I hope this was kind of fun for you guys to kind of check out a bit. And I look forward to seeing your comments in the discussion board for this article. Thanks, guys. I hope you're hanging in there. Have a great day.